Hello everyone, myself Deepa Singh, working as assistant professor in Suresh Gyan Vihar University Pharmacy Department. Today we discuss about stability study and chemical properties. This topic is covered in a pre-formulation study or first unit of Interstate Pharmacy 1 fifth semester subject. So in pre-formulation, we already discussed previous topic. And today we discuss about stability study, how we perform this stability study on chemical properties, including a stability study and different type of stability study according to different type of formulation and which stability is suitable for particular formulation and how we perform this stability study and how this stability study perform and judge your formulation is good or not for particular formulation and according to this stability study you also decide the expiry date and in this stability study topic we start. Before starting this topic you know about basic object. And in this basic object, first is stability study, means definition of stability study or different type of stability study, how you perform and what is stability study. And with the help of the study or stability study, which you include, or next is drug excipient interaction. And you know that different type of formulation contain different excipient and you use different type of excipient. So with the help of this stability study, you can also determine drug excipient interaction or compatibility between drug and excipient, or maybe excipient interaction, because if this interaction is observed, then there may be chances that your drug is not good and good formulation is not formulated, and a certain type of problem may be shown or observed during maybe storage or during administration. So before starting a formulation or finalize your formulation, you should assure about these drug excipient interaction, or maybe drug interaction or drug excipient and excipient also interaction takes place. And next is chemical properties, different nature of that particular excipient or their drug. Means in this you check all chemical property or chemical nature of your drug as well as excipient. Not only excipient chemical property you check, but also check chemical property of your drug also. And in this you check or completely assure about the chemical nature of your drug as well as excipient. Next is stability study at different stages where stability studies require during the pre-formulation or during formulation or maybe after formulation. So which stages is required this stability study or different stages for stability study, and how you perform the stability at various stages. All these you covered in this stability study topic. So now we discuss about first stability or definition of stability. What is this stability? Mainly all type of formulation required this stability study, because with the help of this stability study you finalize your formulation or check the expiry date, how much stay longer, stay good formulation or maybe safe formulation. In this stability study provide evidence on how the quality of a drug substance or drug product vary with time under the influence of variety of environmental factors such as temperature, humidity, and light. Means with the help of this study, you also check that drug substance or your drug product or maybe your formulation may be influenced or not, or maybe show certain changes by the presence of environmental condition or by the changes of environmental condition. This environmental condition may be temperature, humidity, or light. Because you can see that a certain type of formulation are good during the storage, or it reaches to particular time period when you store up to their expiry date. But when you store in a improper condition or not according to required condition, and to expose at that particular environment, this is not good for formulation, then it get easily degrade or show fast expiry. Because it's not appropriate for that formulation. Suppose certain formulation already see that is stored in the dark and away from sunlight. In this type of formulation, if you expose or store on contact with sunlight, there are chances that it easily degrade or maybe do not stable for particular time period. And then this presence of humidity is also affect your formulation. Suppose your formulation is required dry form and you store in a certain humid condition or certain moist condition, and your formulation easily uptake that moisture, so it can easily degrade or maybe change the state. Suppose your formulation in a solid state, but you store your formulation at humid condition or moist condition, then it take moisture of surrounding environmental moisture, and it converted into liquid formulation within a solid semi-solid formulation, and it's not in the original state. So this humidity, temperature, and light is very responsible factor, environmental factor to affect your formulation. So you can check these factors by the help of the stability study, which parameter is suitable for your formulation, which temperature or humidity or light condition is required for your formulation. And after this stability study, mention this condition on the label and give my directions. Next is establish a retest period for the drug substance or shelf life for drug product and recommend a storage condition. You can also perform retest or retesting stability. And by the help of this retest, you perform or check the shelf life of particular drug. And with the help of retest, you recommend that this formulation is good for a particular storage condition or not. And after checking this storage condition, decide the shelf life or their particular expiry date. Next is physical, chemical, and biological, microbiological changes might affect the efficacy, security of the final product. Not only certain environmental factors, but also certain physical, chemical, as well as microbial changes also affect your formulation or maybe change this efficacy and security of your final product. And suppose you do not take a proper physical size drug or physical size excipient, or certain chemicals which are not compatible with each other, then in this case, your formulation may also show certain problems. And if certain microbial or bacteria are present in your formulation or maybe on your container, then it also change efficacy and safety of your formulation and show certain problem on your final formulation. Maybe it get easy degrade or show certain efficacy problem on your formulation. So before finalize your formulation, completely assure about your formulation. Not only check for certain environmental condition, but also physical, chemical, microbial condition is also required for checking efficacy and security of your final product. Next, in this stability study is the extent to which the product retains for its period of storage and use and its shelf life. The same property that is possesses the time of manufacturing. Means stability study, you can say that it's a condition in this during formulation or after formulation, it shows certain specific stability or certain good quality, and this quality is remain stable for their shelf life or during the storage, maybe 2 years, 3 years. We do not show any degradation or any changes of that particular property, and it's maybe it remain for their storage condition as well as during manufacturing. Then stability to the product remains for this period of storage, during storage or maybe after manufacturing to a particular shelf life, it is remains same or showing same property, same to that particular manufacturing time. And one of the principles of dosage form design is to assure that chemical integrity of the drug and substance is maintained during the unstable life of the product. And you can say that one of the principles of the stability study is that assure that chemical integrity of the drug, and you check that chemical integrity of drug is proper or not for that particular drug and maintained during the stable or shelf life of the product. Next is three type of mainly stability studies takes place. First is chemical stability, second is physical stability, then biological stability. These are the stability which mainly require to check a particular formulation is good or not for required condition, or maybe for certain chemicals affect or interact with your formulation or not. And in this chemical stability study, each active ingredient remains chemical integrity within the specified limit. Means during formulation or maybe after formulation, each ingredient, maybe excipient or maybe your drug, are stable condition or maybe remain same condition for a specified time limit, maybe up to the expiry date, and it do not change their property or do not change the specific condition. If this particular excipient or drug get changed, then it may be chances that your formulation also change or it shows certain problem or certain instability problem. And so your formulation should be stable or should have same integrity of each and every excipient as well as your drug. Next, in physical type of stability, the original physical property including appearance, taste, color, and odor are retained same. Means after formulating or during formulating, it do not change their appearance, taste, color, and odor. It should be remains same for that particular time. Suppose you manufacture or formulate transferred formulation, but during storage after 6 to 1 month or maybe 1 up to 1 year, it changes their color and it converted into orange or maybe red or any other color. So this type of problem should not occur during your formulation or after your formulation. And certain taste changes is also observed during the storage condition, or maybe color and odor also change. So your formulation should be remain stable for a particular time period or up to the expiry date. It do not change. And in biological conditions, stability is retained, no microbial growth. I mean, certain bacteria or certain microbes are present on your formulation. So in this condition, your formulation is also changed or show certain problem in your formulation manufacturing. So this problem is overcome, and before compiling a formulation or before finalizing your formulation, you unsure about their stability and you properly manufacture or formulate your formulation in good condition, which are sterilized condition or free from microbes or any bacteria. And not only formulation but also cross check your packaging material. This packaging material is don't have any bacteria or microbes. If this bacterial microbe is present, then it may interact with your formulation and show certain stability problem or maybe degrade your formulation. So not only your formulation ingredient including drug and excipient are in sterilized condition, but also packaging material as well as your plant condition is also free from microbes or free from bacteria, means it's a sterilized condition. So if you take all these precautions in this condition, your formulation is good quality formulation or show free from microbes or show stability for particular time limit until their expiry date. Next, in a stability study are performed on drug substance or maybe on a drug product. In drug substance, the unformulated drug substance that may subsequently formulated with excipient produce a dosage form. Drug substance means maybe your excipient, maybe your drug or individual ingredient. So before starting your formulation, you also check drug substance stability individually. And in this mainly you check subsequently with particular time limit or check the interaction, maybe excipient or drug interaction or drug interaction or excipient interaction. This all drug substance required for your formulation should be free from microbes or should be in a stable condition. You assure all this property. Next is drug product. In drug product, the dosage form in the final immediate packing included for market. Drug product means after formulation or before coming in a market, you should assure about all formulation or your final immediate formulation should be in a sterile condition or the packaging material should be in a sterile condition. So dosage form in the final immediate packing immediate for marketing, you should assure about your stability study. It should be free from microbe and should be in a stable form. Control and a documented determination of acceptable changes of drug product and drug substance. In this, there are certain acceptable changes or certain drug product show problem. In this, you check the stability study for control limit and maintain particular documentation, observe all reading and record in a certain document and mention these are according to requirement or not. If certain problems are occur, you verify the record. So after this compiling record, you can easily check which step should this problem or so you do not maybe start or complete batch or maybe maintain or changes on that particular batch after recognize that particular step of your manufacturing process. Next are what are the changes are majorly you observe on your formulation. In this, physical change, chemical change, and microbial change. You know that physical change means maybe the color, odor, or taste changes. And in this physical changes classes, it again further divided in different parts like appearance. Appearance means a formulation change their appearance. Suppose it's in a transient or solid state, then it will convert into semi-solid or liquid, and the appearance is also not same as previous one. Next is melting point. Melting point is also very important. Your formulation may be changed. Your melting point, suppose melting point of that particular formulation or drug is 48, but during physical stability problem or physical changes, it may be enhanced or decrease. Or suppose example if SN58, and during storage or physical changes, it changes up to 75 or 72 or more than this. So this melting point is also you check before finalize the formulation, which is melting point of that particular formulation or particular drug, and after storage is stable or not. Next is color and clarity of the solution. In this color and clarity of solution is not for all type of formulation, but certain liquid formulation or semi-solid formulation is required this test. Suppose you're preparing gel, and gel is in transit clear solution. In this condition, so you check that clarity and color of the solution, and you prepare semi-solid gel-like formulation. In this condition, you assure that the clarity of your formulation should be clear. And suppose you prepare transparent gel, and during storage it changes color and it converted to maybe creamy or orange color. And this is mainly about the physical changes. So you know all this physical parameter, and check clarity and color of the solution. And not only semi-solid formulation, but in your liquid formulation, this clarity testing is also required with the help of black and white background to check there's certain bacteria is present or not, or certain foreign particles may also present in your formulation, to assure that your formulation should be in a clear form, free from micro or any foreign particle. And there's certain type of method is also available for clarifying this clarity and color of the solution. Next is moisture content. Moisture content is also for very depending on type of the formulation. If suppose you prepare tablet, and it should have sufficient moisture, because if it's not have sufficient moisture, then it form too much hard or easily get break. So in this condition, you should have check the moisture of particular tablet or particular formulation. And if your tablet contain more moisture, then there are chances that it can easily degrade. During this condition, you assure that how much moisture is required for your formulation. So you don't say that your formulation should be contain moisture, but it should be in required amount. If it's not in required amount, then it may show certain problem or maybe show certain stability problem. Suppose your tablet have more moisture, then it can easily degrade or show certain bacterial growth. If it's not have sufficient moisture, then it can show more hardness or maybe show dry tablet or do not disintegrate or absorb particular amount of the tablet. Next is crystal modification like polymorphism. Polymorphism means more than one type of the crystal. Suppose you require polymorphism formulation or polymorphism drug, then in this condition you change your crystal form or maybe convert it into polymorphism, so you can formulate good quality of the formulation. Next is particle size. Particle size is also very important factor. And this you reduce particle size for better absorption or better distribution. But in certain cases, this small particle size is not required because it may be because larger particle size show good control bliss process, and this particle size is decide according to type of the formulation. And a certain type or majority of the formulation is required smaller type of particle size, because smaller particle size, larger the surface area and more the absorption rate. And next is chemical changes. In chemical changes, mainly you check the increase in degradation or degrees of the assay, means how much drug is degrade, a particular amount of the drug is degrade. Or assay means you just confirm your formulation should have sufficient dose or not. Suppose you formulated 500 mg drug for your formulation, but it do not show all 500 mg drug effect. It show only 300 mg. So in this condition, you perform assay test and assure that all drugs are in a particular required condition or not. Next is microbiological change. In this, due to certain bacteria or microbes, certain changes takes place. And in this, next is chemical and degradation stability study. Chemical degradation stability is also further divided into different parts like hydrolysis, oxidation, reduction, decarboxylation, and photolysis. Hydrolysis, we already know about the breakdown of the particle or removal of a water molecule. Oxidation means there is a certain oxidation or oxidized process takes place in a formulation. Reduction means particle size, well, not particle size, but removal or reduction of the condition. And decarboxylation means removal of carboxylic group. Photolysis means mainly due to presence of light or sunlight, this photolysis takes place. Now we discuss about stability study for solution and solid state stability. In solution stability, the decomposition of a drug occurs through hydrolysis, oxidation, and photolysis. These are the 3 types for solution stability study. In hydrolysis, example like anesthetic, vitamins, etc. And in this example is ester hydrolysis like RCOR plus H plus OH is the ester group, and when react with H2O, this ester converted into acid and alcohol means RCOR is the ester group, and H plus OH is water molecule, and with this water molecule react with ester, it forms acid and alcohol, and it show hydrolysis reaction because the removal of water molecule or breakdown of the water molecule due to presence of this water molecule, it converted into acid and alcohol. Next is amide hydrolysis. In amide hydrolysis, there is amide group RCO and HR. And this amide is react with water, means RCO and HR react with water, and the presence of water form acid and amine. Again, in this hydrolysis takes place or removal of water molecule takes place like RCOH acid form and H2 and R amine is formed. Next is oxidation reaction. In oxidation reaction, used to evaluate the stability of pharmaceutical preparation. In this, you just confirm that your pharmaceutical preparation are good or not, or properly showing stability or not. In this, mainly example of drugs like steroids, vitamins, antibiotics, and epinephrine. And oxidation like auto-oxidation maybe takes place due to environmental condition or certain environmental changes. In this, material plus molecular oxygen show homolytic fission, and material and molecular oxygen show homolytic fission, then it will release free radical or maybe produce free radical. Means suppose your material contains certain molecular oxygen or certain amount of oxygen. Due to the presence of this oxygen, there is a homolytic fission is occur, and this homolytic fission is mainly responsible for releasing or producing free radicals. And this process is known as oxidation, and this is also known as oxidation because it takes itself due to presence of certain environmental condition. And next is oxygen sensitivity is measured by coupling air to the compound or adding hydrogen peroxide. And this oxidation or auto-oxidation maybe shows certain sensitivity or oxygen sensitivity is observed, and you can measure by the bubbling through air to compound or adding hydrogen peroxide. When you use hydrogen peroxide or bubbling through the compound, then you can also measure this oxygen sensitivity, means how much oxygen is sensitive for your formulation or how much oxygen is responsible for degradation of your product. Next is photolysis. Photolysis, you know that this is a factor that is affect your formulation by the presence of the sunlight or by the presence of light. In this, maybe you can say that pharmaceutical compound exposed to UV light, and due to this UV light undergo degradation reaction, and it absorbs the radiant energy and responsible for degradation of your formulation. Means suppose you can say that you're taking certain formulation and your formulation is a tablet capsule and or any type of the formulation, and you store on the exposure of the sunlight or the presence of the sunlight. And due to this presence of the sunlight or exposure of the sunlight, in certain radiant energy is absorbed or energy is absorbed by your formulation. And due to absorption of this sunlight or UV light, the formulation shows certain changes, and these changes are responsible for degradation of the product or changing the stability of the formulation. So in this condition, you work on this problem You should assure that your formulation in a good storage condition, or it should be stored away from sunlight or directly away from light. Next is solid state stability study. In the solid state, first is objective, identification of stable condition, identification of compatible excipient. In solid state stability, first starting performing the stability study for solid state, you should assure that your storage condition is good for your formulation, plus your compatible excipient are selected. Suppose you selected improper excipient or not compatible excipient, then in this formulation, there may be chance that the formulation complete degrade or not suitable for particular storage condition or particular time, because it contains incompatible or certain drug or excipient interaction maybe takes place. So you assure that all excipients are compatible or good quality for your formulation, and this is assured by formulating or performing certain pre-formulation study. Means you perform drug-drug interaction, or maybe drug-drug, not drug-drug excipient interaction, excipient interaction, or just compatibility check of particular excipient with drug. And in this, you can also perform the solid state stability depending on temperature, light, humidity, and polymorphic changes and oxidation. So in this condition, you also assure that all these changes are not or present or not in your formulation. If you store your formulation in particular temperature, light, humid condition, or so in this condition, there is no polymorphic changes are take place or oxidation is not takes place. Next is drug excipient compatibility. In drug excipient compatibility, you can say that compatibility of a test play a very important role in pre-formulation study of the oral dosage form. And incompatibility in the dosage form can result in any of the following changes. Change in organoleptic property means you can say that if you're formulating good compatible formulation or very compatible formulation, this is mainly condition for oral solid dosage form. And if your formulation is 100% compatible, then no problem is observed, and your formulation should be stable for a particular time limit or up to the shelf life. But your formulation shows certain incompatibility problem or certain problem. Maybe there is a drug excipient interaction or any other incompatibility problem. In this case, you can see that certain observed condition are you see. First is change in organoleptic property. Your formulation may be changed their organoleptic property. Organoleptic property means maybe taste, odor, color. These type of property are changes. And next is change in dissolution performance. Dissolution rate is also changed. Maybe it's a more fast dissolution or maybe slow release dissolution. Suppose you prepare fast disintegration tablet, but due to this incompatibility issue, it shows slow release or maybe delay release of the formulation. Next is physical form conversion. In physical form conversion, you can say that it can change the state, maybe solid to liquid or liquid to solid formulation. So you can also assure that the formulation should be in a compatible state and it do not change the physical form. Suppose you store your formulation or your tablet in a certain humid condition, and due to taking this humid or maybe due to the certain environmental changes, it take moisture and convert their state, and after solid state it convert into liquid or maybe in semi-solid state. And decrease in potency. Required amount of a drug effect is not observed and it is not potent to show that sufficient amount of the effect. So during this incompatibility study or incompatibility of a formulation, potency of a drug or efficacy of a drug is also changed, or it may be show decrease amount or increase amount. So you can store your formulation to form compatible formulation. Next method. In this method, first you take drug and excipient, and concentration and ratio of this drug and excipient is 1:1. And suppose you are taking 10 gram of your drug, and in this condition you also take 10 gram of your excipient also. Both are in a similar amount or similar concentration. And then powder of samples dispersed into the glass. And then store at a particular temperature, mainly 50 degrees Celsius, and analyze in the immersion study include measuring the critical micellar concentration of the formulation. And for oral use preparation, compatibility of ingredient like ethanol, glycerin, syrup, sucrose, buffer, and preservative. Suppose you evaluating your stability study of particular formulation. In this, first you take drug and excipient, and both drug and excipient are in a similar amount and similar ratio. And after this, you disperse in glass ampoule both drug and excipient. And there's a two ampoule. One is for sample and one is for water. And after storing in this particular condition, mainly you prefer 50 degrees Celsius temperature. And after storing this 50 degrees Celsius temperature, you analyze your formulation. Suppose you testing this test for emulsion, then you are sure that critical micellar concentration, how much concentration of CMC is changed or maybe remain same. And for oral preparation, mainly check the compatibility with an ingredient or with excipient like ethanol, glycerin, syrup, and sucrose, buffer, and preservative type of excipient. You check. This is a diagram how you check your stability study or interaction with excipient with the help of certain equipment or machines. Suppose you are taking drug and excipient 50% mixture. And after taking this drug and excipient 50% mixture, you check DSC, differential scanning colorimetry. This is a technique. In this, you check compatibility between the drug and excipient, and you can also say that the drug are compatible with your formulation or not with the help of this DSC technique. And after performing this DSC, you can observe the result. If no interaction takes place, then recommended that particular excipient is good for your formulation. If there's an interaction is observed or an interaction is seen in the excipient interaction, then you say that you perform TLC. TLC is a thin layer chromatography, or you can say that if your formulation is recommended, or sorry, your excipient is more recommended, and then you can show that this is for final formulation. Means first you take drug and excipient. This drug and excipient have a 50% mixture concentration, and then perform DSC. After performing DSC, if there's no interaction is observed, then you recommend it to excipient. And if there's an interaction is observed, then you perform again second test, that is TLC, thin layer chromatography. If thin layer chromatography is significant or maybe show no breaking of any compound, then you can recommend your formula excipient. If there's a certain problem is observed, then you change your excipient and use alternate excipient. There may be some chances that drug excipient starting stage, you can change your excipient also or use alternate excipient. If your excipient is good, you can recommend your excipient for your formulation. If this excipient again showing some problem or by the TLC method, then you can change your excipient or use certain alternate excipient. Next is stability study at different stages. In this, first is selection of sample. In selection of sample may be API, excipient, or bank. This may be you check active pharmaceutical ingredient, means a drug, or maybe excipient, or maybe in a batches from API and your excipient. So this selection of excipient, after this scope. Scope in this means which parameter you check. In this, first is appearance. Second is appropriate physicochemical parameter. Second is assay and degradation product. Means any changes are observed or not in your formulation. Then appropriate physical and chemical parameter. Certain physical or chemical changes are observed. Then assay and degradation of the product. Assay means particular amount of drug is show effect or not in your formulation. And this study is mainly for up to 3 months. If you perform this study for 3 months, maybe every month or maybe at particular interval of time, but we perform at least up to 3 months. And scope, second test is perform solubility profile, hygroscopicity, thermal stability. In thermal stability, mainly perform melting point, polymorphism, and chemical stability. In this, one batch is required, and this study is also performed for 3 months. Means you can say that if you are testing solubility, your profile, hygroscopicity, and thermal stability, and melting point and polymorphism, and maybe for certain chemical changes you see. And for this condition, you will perform a one batch stability study for up to 3 months or maybe particular time period. And next is to perform stability study for at least 36 months, means up to 3 years. And this stability study mainly performed for determination of expiry date, means how much drug stable for a particular formulation or particular time period. And with the help of this test, to check expiry date of the formulation, determination of preliminary significant specification. Preliminary specification, so starting specification also check. Release of clinical batches, monitoring of a sample during the clinical phases, definition of the storage condition, definition of test for registration of stability. And this test will mainly perform for 36 months, means up to 3 years, and they check this study for maybe one month interval, or maybe 6 month interval, or maybe after 1 year you perform this, or a 3 month interval, and after 1 year you perform interval of 6 months, or maybe from 1 year, but you perform at least 36 months or up to 3 years. Next is testing score for solid dosage form. And solid dosage form, you know that main tablet and capsule are we covered. And this before starting or before performing this tablet or capsule or solid dosage form tablet study, you check certain parameters like physical, chemical property, microbial property, and container closure system property. Container closure system means this, you can say that you assure that your tablet or capsule should be in a proper appearance, and you do not change their appearance during storage or maybe particular time period. Elasticity should be maintained. Suppose in capsule we use polymer and certain elasticity is present in your capsule, then in this case this elasticity do not change or maybe do not reduce. Mean mass means the mass or weight of the tablet or capsule that remains stable or remains same until the expiry date. Next is moisture content. In tablet and capsule, certain amount of moisture is present, and you can assure that this moisture do not change. And this moisture is in required amount. Required amount. If more moisture is present, maybe the chance that it degrade your formulation. Next is hardness. If the tablet is in good hardness, then it should have good wear and tear property, and it do not degrade or maybe do not break during storage condition or during transportation. So in this, you assure the tablet should have a sufficient hardness, and disintegration means breaking of a tablet into small particles. So in this condition, you assure that your tablet should disintegrate properly or break down in a uniform or proper particle or small size particle. Next is resolution rate. Resolution rate is also important for both tablet and capsule. And this after disintegration, dissolution rate takes place. In dissolution, you can check that how much drug release in your formulation for particular effect, or how much percent drug release in your formulation. Next is chemical property. In chemical property, you check acid. With the help of this acid, you check that all amount or all concentration of drug are responsible for effect showing a particular formulation. Suppose you are taking 500 mg tablet, then you check that all this 500 mg is present or not in your tablet. Maybe it is less or more than 500 mg. So in this condition, there is certain limit or certain variation is included in pharmacopoeia, and you cross-check or verify this limit with the particular standard books. Next is degradation. Degradation means changes of certain property or degradation of the formulation. And in this, you assure that your formulation, tablet and capsule both are in a good condition and do not degrade easily. Microbial property means certain bacterial microbes that affect your formulation. And container closure means functionality test, maybe extraction from a blister. You know that the tablet or capsule are packed in different type of packaging material like blister packaging, strip packaging, or alloy packaging. In this, we assure that all type of packaging material, foil material are in a good condition and do not interact with your formulation, and it should store your formulation in this good time period or maybe good condition. So you can check this container closure condition also, or interaction with drug or packaging material also. Next is testing score for liquid form. And in liquid form, mainly you can say that you perform this test for oral liquid form like syrup, elixir, suspension, this type of formulation. In this, you require physical chemical property like pH. The formulation should have a good or required pH. Suppose the required formulation effect show in stomach, then it should be in an acidic pH, not in a basic pH. Or for intestine, it should be an alkaline pH. Next is color and clarity of the solution. It should be clear or don't contain any foreign particle or any microbes in your formulation, and you perform this test by the black and white background. And color of your formulation will remain same for a particular time period. It do not change. Next is particle size distribution. This is mainly for suspension only. And in this, you check that your particle size present in a particle or present in a suspension should be in a uniform form or should be in a small form, and it do not change or do not form aggregate. When it get form aggregate, then it do not show particular desired effect. So particle size in uniform distribution, or it should be uniform in size, and it do not form aggregate during storage. Next is assay. In chemical property, in assay you can assure that a specific amount of drug is present or not in your formulation. Suppose you are preparing syrup and concentration of the drug is 1 gram in a 50 ml syrup. So in this condition, we check that that particular 50 ml container should have a sufficient or required amount of drug concentration or not. Next is degradation product. In degradation product, you check that the product is degraded or not. And the degradation of preservative, certain preservatives is also responsible for degradation. You select good preservative which helps to preserve your formulation, not to degrade the particular formulation. Next is contain antioxidant. You can use certain antioxidant for preservation of oxidation reaction. In this condition, you select proper good condition antioxidant. Next is microbiological property or microbial property. In this scene, similar to solid acetone. In this, you check container or formulation excipient or your packaging material or appropriate or not for your formulation. Next is testing scope for liquid form for injection, and this is for parenteral formulation because injections are parenteral formulation. In this parenteral formulation or for injection, more care is required or more sterile condition is required because these formulations are very sensitive and the formulation is directly administered in your blood or maybe in your vein and directly just to your blood circulation. So this formulation should be required more and more sterilized condition or more and more caution are required. And in this parental physiochemical properties means pH, the formulation should be required pH. If it's not a required pH, then person maybe suffer certain problem or up to maybe reaches to death. And so you perform your formulation to required pH. Then loss on weight, maybe certain formulation show weight changes or maybe loss of the weight is observed due to improper storage condition or environmental condition changes. Next is color and clarity of the solution. The formulation should not contain single or maybe no bacteria or foreign particle, or it should be in a proper sterilized condition or free from microbes, and a clear solution. You should perform all the tests required for this color and clarity testing. Next is chemical property. In chemical property, first is assay. You check that all required amount or added amount of drug is present or not in your formulation. Next is degradation of the product. Degradation preservative. If required, then you use preservative. Otherwise, in certain parenteral or large volume parenterals, no preservative is required. And if you use preservative unnecessary, then there may be chances that it can degrade your formulation. Next is contain the antioxidant. You use antioxidant if required. And before selecting this antioxidant, you assure that your antioxidant are good quality. Next is container closure property, same for previous one. In this, you check that all container or required storage material are good or not for your formulation. Next is testing scope for semi-liquid form. In semi-liquid, mainly your cream and gels are covered. And in this, physical and chemical property, mainly appearance, odor, homogeneity, and consistency. The appearance should be remain same, and no odor is observed during storage. Homogeneous means if do not separate their phase, the oil and water phase, and it should be a homogeneous or uniform phase, and should have a consistency for applying It should have sufficient consistency, like semi-solid dosage form. It should not be changed their state or maybe converted semi-solid to liquid. Should have a sufficient consistency up to their particular time period. Loss on weight and viscosity. Viscosity is also maintained, and loss of the weight is also not observed in your formulation. Next is contained uniformity within the contained. Means contained or your formulation should be in uniform form and not show any changes during storage or within the container. Next is chemical property. In chemical property assay, to assure that all amount or added amount of drug is present or not in your formulation. Next is degradation product and preservatives. In this one, in this you assure that preservative do not degrade your formulation or select proper preservative if needed. Contain preservative, some type of contain or excipient may react with preservative. In this, check that contain preservative interactions not take place. Degradation or contain antioxidant. If you are using antioxidant, then you assure that this particular antioxidant is good for your formulation. Next test is microbial property. In this, check that certain microbe or bacteria present or not in your formulation. Then container exposure system property. In this, you assure that your container or packaging material should be in a good quality or do not show any interaction with your formulation. This is all about stability study of formulation according to different type of formulation. So after completing this topic or after completing this PPT, you should know about all the parameters regarding a stability study, including different type of formulation, stability study for semi-solid, liquid or solid dosage form, and including parenteral dosage form, which parameter you check or which parameter is required to changes if certain problem is occur, and how you perform this stability study. All this you check in this PPT, and you know about definition of stability and what is stability study, and drug excipient interaction, how you select particular excipient, and how you check drug interaction takes place or not, and chemical property and stability study at different stages, which stages require for stability study, and how much time period is required to perform this stability study. Next is reference part. In reference, first is Ansel Pharmaceutical Research Form, Drug Delivery System, 8th edition by Lord D. Nicholas, Chief of which Howard C. Ansel, published by publication PPT Limited. Pharmaceutical Preformulation by J.T. Cartesian, published by Technomical Publishing Co. Next is Textbook of Physical Pharmaceutics by C.V.C. Subramanian, published by Balla. Theory Practice of Industrial Pharmacy by Leon and Lackman Haber, Lieberman Joseph, L. King, 3rd edition, published by Bargesi Publishing House. Fifth one is Martin, Physical Pharmacy, Pharmaceutical Science, 5th edition by Patrick J. Senko, published by Liverpool, William and Pilgrim. And sixth one is Pharmaceutical Dosage Form Tablet, Volume 1, edited by Herbert Lieberman, Leon and Leishman, published by Martian and Becker. These all references are good. You can use any one reference or maybe a combination of these above books. And next part is question part. In first question, all oral solid dosage form show greatest chemical and microbial stability. B is more impurity. C is more administration problem. D is more irritation. The second question, drug that are sensitive to oxygen may require encapsulation or coating. Next, B is binder. C is diluent, and D is sugar coating. Next is the third one is aspirant is artificial sweetener as a main disadvantages carcinogenic, and B is metallic aftertaste, and C is bitter aftertaste. D is hygroscopic in nature. And fourth question is why emulsion dosage form and lipophilic drug pattern. Then A is easy manufacturing. B is the higher stability of a drug in emulsion form. C is presented a large surface area of oil to the GIT for absorption of the drug. D is a fast clearance from the plasma. Fifth question is from strength is tested using the instrument like biometer, and all mentioned. C. Sixth question is in the stability of the emulsion, which imparted stability and stability step should be prevented by breaking, persistence, flocculation, and framing. Next, seventh question is Brownian movement of particle in suspension caused by A is assist sedimentation, B is promote sedimentation, C is prevent sedimentation, and D is increase sedimentation rate. And in eighth question, the point of interest for development of successful capsule dosage form are first is physical and chemical stability, there is a fill composition, C is shell material, and D is all mentioned. And ninth question is in process tests are done to reduce the cost, to consumer acceptance, to minimize the risk of product failure, and D is none of mentioned. 10th question, FTIR assay and limit test are mandatory for the testing of finished product, raw material, in-process material, none of the mentioned. All these are the questions regarding a stability study on this particular topic. So you can solve this question. And thank you so much for watching and listening this video. Thank you so much, everyone.